Hi, we are Group 4 and we will be talking about medically significant viral disease, specifically diseases caused by Rhabdovirus. But first, let me introduce our group. I am Sheanina de Guzman and together with me is Miss Aubrey Balmaceda, Miss Millicent Lanzuela, Miss Marge Molina, and Miss Lineah Untalan. We will be talking about the following: characteristics of this virus, its mode of transmission, epidemiology, pathogenesis, immune responses elicited by the host, and lastly is the prevention and treatment. Now let's start with its characteristics. Rabies virus is a medically significant virus under family Rhabdoviridae that contains a negative-stranded RNA genome. These bullet-shaped envelope viruses are very stable to drying that have an icosahedral capsid symmetry. It is highly potent, which connotes a 100% mortality rate caused by the virus. Family Rhabdoviridae, specifically under Lissa virus, is one of the oldest and the most feared viruses in the history of mankind.
Transmission of rabies virus. The transmission mode of the rabies virus to humans occurs only horizontally, with the minimal requirement of saliva deposition containing the virus. This may take place in 4 ways. First, bite of a rabid animal, where usually the principal vectors are dogs, which serves as the inoculation entry of the virus, in which all individuals are susceptible but to greatly varying degrees. This manner of transmission accounts for most of the cases reported of human rabies cases worldwide. Second, contamination of an open wound via licks and scratches. Third, contamination of an intact mucosa, such as the mucous membrane of eyes, nose, mouth, and genitalia with the saliva of an infected animal. And fourth, inhalation of respiratory droplets containing highly concentrated live rabies virus. The latter 3 are collectively known as non-bite exposures, which attribute also in causing rabies to humans, however rare. Epidemiology of rabies virus. Rabies virus infection and disease occur in nearly all countries without the exception of our country, and it is endemic, which remains to be a public health concern. Where the epidemiological study conducted by the Department of Health of the Philippines from January 1, 2014 to June 30, 2018 revealed that 1,176 total cases were reported with an average of 258 cases annually, having a fatality rate of almost 100%. From this population, 73% were males with ages ranging from 3 years old to 87 years old. 92% of the cases surveyed were due to suspected rabid dogs, followed by cats amounting to 3%, where 49% were domesticated and 38% were stray animals.
The pathogenicity of the rabies virus is a multigenic trait involving various RABV-encoded proteins and transcriptional elements, an RNA-dependent RNA polymerase or L, a nucleoprotein or N, a phosphorylated protein or P, a matrix protein or M, and an external surface glycoprotein or G. The N, L, and P proteins collectively form the genomic RNA called the ribonucleoprotein complex or RNP. The G protein is the primary determinant of the pathogenesis of rabies virus by facilitating rapid virus entry and immediate transsynaptic spread and regulation of the rate of virus replication. Also, P protein is a vital factor in the retrograde axonal transport of the virus. The rabies virus is divided into 2 forms according to their ability to invade the central nervous system. Tissue culture adapted RABV strains have no or limited ability to invade the CNS, whereas street RABV strains are highly neuroinvasive. Street viruses are those isolated from animals and humans, while tissue culture adapted viruses or fixed strains are those repeatedly passaged intracerebrally so that it becomes highly adapted to brain tissue and then able to replicate in extraneural tissues. The susceptibility to infection and the incubation period is influenced by the virulence of the virus strain, the amount of inoculum, and the location and severity of the bite. A higher attack rate and shorter incubation period in individuals bitten on the face or head are established, whereas the lowest mortality occurs in those bitten on the leg. The incubation of rabies virus in humans and animals usually lasts 20 to 90 days or shorter, depending on the distance of the bite from the central nervous system. Following inoculation into subcutaneous tissues and muscles, the virus replicates in the striated and connective tissue and binds to one of the 3 cell surface receptors. Neural cell adhesion molecule or NCAM, P75 neurotrophin receptor or P75-NTR, and nicotinic acetylcholine receptor at the neuromuscular junction. Then, the virus is internalized via clathrin-dependent receptor-mediated endocytosis. The RABV-containing endosome enters the axon terminals and directly travels to the central nervous system via retrograde axonal transport at a rate of 12 to 100 mm per day, where it extensively replicates. After forming an endosomal compartment, the low pH environment induces a conformational change in the G protein, triggering the fusion of the viral and endosomal membrane, subsequently releasing the RNP into the cytosol. Viral replication takes place and pathologic effects such as inflammation manifest affecting neuronal physiology. There, via anterograde axonal flow, it spreads into peripheral organs such as the salivary glands, cornea, and kidneys. Following the systemic dissemination of the virus and clinical onset, shedding of the virus takes place, typically in saliva, completing the infection cycle of rabies virus.
The infection of rabies virus manifests diverse signs and symptoms during incubation and prodromal period, which is typically about 2 to 3 months but may vary from 1 week to 1 year, dependent upon factors such as the location of virus entry and viral load. These symptoms are usually non-specific like fatigue, fever with pain and unusual tingling, general malaise, paresthesia at the wound site, or implies an association of the gastrointestinal system, such as abdominal pain, anorexia, diarrhea, dysphagia, nausea, and vomiting, respiratory system like cough, dyspnea, and sore throat, or the central nervous system where anxiety, apprehension, irritability, headache, nervousness, and vertigo may occur. This follows the acute neurologic period where a progressive and fatal inflammation of the brain and spinal cord develops, which is also an objective sign of dysfunctionality in the central nervous system as the virus circulates. Consequently, this leads to the formation of rabies disease, which may occur in 2 forms. First, furious rabies, which results in symptoms of aerophobia, aggressive behavior, hallucinations, hydrophobia, hyperactivity, insomnia, and spasms of the muscles in the larynx and throat that are usually triggered by breeze and water. After a few days, the cardiorespiratory arrest usually happens to cause death. Second, paralytic rabies. As the name suggests, the patient experiences paralysis at the site of primary infection, that is, at the site of the bite or scratch, which gradually advances to the muscles. A coma slowly develops and eventually, death takes place. This form of rabies accounts for about 20% of the total number of human cases and is considered less detrimental, however, usually with a longer course than the furious form. On the other note, several signs including fever, focal and generalized convulsions, hypersalivations, hyperventilation, muscle fasciculations, nuchal rigidity, and paresthesia may be observed in both forms of the disease. The chances of getting rabies are exponentially higher when the following risk factors are involved. The following list is completely adopted from the World Health Organization. The diagnosis of human rabies can be straightforward, granted that the patient exhibits the full spectrum of the linked signs and symptoms of the said disease, and especially if the history of an animal bite or scratch exposure is evident. Otherwise, the establishment of conclusive diagnosis necessitates a rapid laboratory assessment and the detection of rabies antibody, antigen, rabies virus, or viral RNA. This test may be done in 5 ways. First is skin biopsy sample from the nape of the neck, examined either under a microscope for the identification of virus or direct immunofluorescence test for the presence of rabies antigen. Second, saliva collection for the culture and inspection of rabies virus. Third, lumbar puncture to obtain a sample of cerebrospinal fluid for the detection of rabies antibodies along with the serial serum specimens. Lastly, the polymerase chain reaction or PCR technique, which is used to verify the presence of viral RNA in cerebrospinal fluid, saliva, or skin sample.
The host immune response against rabies virus infection involves both the innate immune response and the adaptive immune response. Moving on to the innate immune response, its components are the following. First, pattern recognition receptors of the families Toll-like receptors and retinoic acid inducible gene 1, which are active in the periphery, facilitates the early recognition of rabies virus infection by detecting its RNA immediately after the inoculation of the virus in the skin or muscle by scratches or bites. This consequently activates both the nuclear factor kappa B and type 1 interferon-regulated responses. The uptake of rabies virus into intracellular compartments via endocytosis and phagocytosis is accounted to macrophages, which also plays a role in the virus recognition. Antigen-presenting cells such as dendritic cells function in the activation of naive T cells. The recognition and killing of virus-infected cells and inhibition of viral replication are mediated by natural killer cells, whether directly or with the assistance of cytokines including the IFN alpha, beta, or gamma. RIG1-mediated response in astrocytes, microglia, and neurons is triggered upon progressing of the infection to central nervous system tissues. This induces the classical primary IFN response, which is the activation of nuclear factor kappa B and interferon regulatory factors, especially the IRF3, along with the secondary IFN response, which is the activation of IRF7 and the STATs, both leading to the simultaneous secretion of cytokines, including interleukin 5, 6, 12, and tumor necrosis factor alpha, and chemokines including chemokine ligands 2 to 5, CCL11, CXCL1, and CXCL10. This further promotes the upregulation of major histocompatibility complexes. Now we move on to the adaptive immune response, which is subdivided into cellular and humoral immune response. The cellular immune response is underscored by the recruitment and activation of T cells, specifically CD4 and CD8 T cells, in the infected central nervous system via CXCL10 secretion. This particular chemokine also aids in helper T cells differentiation into Th1 and Th17 cells, which produce IFN gamma and IL-17 respectively. The sensitization of these T cells promotes the combat of the complete clearance of rabies virus infection. The humoral immune response virus elimination is preceded by the rabies virus-specific antibodies production by infiltrating B cells. The neutralization of rabies virus infection has been demonstrated by the potency of complete IgG molecules. IgM antibodies, on the other hand, prevent the circulation and spread of the pathogenic rabies virus to central nervous system from the peripheral sites. Moreover, the differentiation of B cells to long-lived plasma cells and memory cells are provided by the growth and selection signals to B cells by the T follicular helper cells, whether intracellularly or extracellularly. In summary, the interplay of host immune response, including the innate immune elements, cell-mediated immunity, and the virus-specific antibody response, are all essential for the recovery from rabies virus infection. Prevention and control. It has been mentioned earlier that the fatality rate of rabies virus is almost 100%. However, being the most fatal among infectious diseases, it is also 100% preventable. The prevention and control program of the rabies virus to humans that is formulated by the DOH highlights the significance of pre-exposure prophylaxis or PrEP and post-exposure prophylaxis or PEP. Essentially, these are the protocols needed to administer to all individuals who are susceptible to contracting rabies. There are 3 types of rabies vaccines that provide anti-rabies protection. First, human diploid cell rabies vaccine. Second, purified virus cell rabies vaccine. And third, purified chick embryo cell vaccine, which are all prepared via tissue culturing and are intradermally injected on the anterolateral aspect of the thigh in infants or at the deltoid area of each arm in children and adults. These vaccines induce neutralization of the rabies virus via production of T cells. The vaccines for PrEP and PEP are basically the same. However, PrEP is given to an individual who is at high risk of getting rabies, while PEP is given immediately to individuals who are exposed to the rabies virus. This active immune response is disseminated in 7 to 10 days after 5 series of vaccination shots, which may persist for years provided that primary immunization is completed. Passive immunization such as rabies immune globulins or RIG is also combined with the PEP vaccination procedure. This provides the instantaneous availability of neutralizing antibodies at the site of the exposure. There are 2 types of RIG. First, human rabies immune globulin derived from plasma of human donors, and second, highly purified antibody antigen binding fragments produced from equine rabies immune globulin. Other policies directed in the prevention of rabies virus are dog population management, dog vaccination, the foundation of the central database system, health promotion, and responsible pet ownership. All of these movements are needed to be implemented to realize the vision that is rabies-free Philippines. Treatment. Once the signs and symptoms of rabies begin, management and supportive care should center on avoiding intubation, ensuring comfort for the patient, prevention and life support measures, and using sedation, considering the absence of a cure for human rabies and its fatal outcome. Medication may be given any of the following. First, diazepam, which is taken orally every 2 to 4 hours in children and through intravenous therapy or IV at 10 to 15 minute intervals in adults. Second, midazolam, which may be injected intramuscularly, IV, or taken orally every 4 to 8 hours in children and adults. Third, haloperidol decanoate plus diphenhydramine, which may be injected intramuscularly or via IV every hour until the patient is calm in children and adults.